For my senior research project here at Piedmont Governor's School, I studied methods that would reduce bacterial contamination in cosmetic applicators. My name is Sophia Gieber, and I'm a senior at George Washington High School. I'm interested in continuing my education at Virginia Tech with a major in food science. This major will allow me to pursue a career in many different fields, such as nutrition, holistic healthcare, or pre-med. I chose this project because of my interest in how everyday products impact our skin and to build awareness around how the products and tools used on our skin affects our skin health. Cosmetics, from the tools and applicators to the products themselves, are a major vessel for transferring bacteria to our skin. While conducting preliminary research, I learned that there is little to no research on how to prevent the spread of bacteria through cosmetics. I also discovered that there is a lack of education surrounding cosmetic hygiene in the cosmetic industry. Through the current study, I hope to raise awareness around the importance of good hygiene and establish ways the spread of bacteria through cosmetics can be limited and hopefully prevented. Most of the studies I came across identified or tested similar strains of bacteria. This being because these are the most common strains found in the environments the studies took place in, such as medical or dental, and are the most common strains found on the skin of humans and cause the most illnesses in humans. Pseudomonas aeruginosa is typically found in hospitals and healthcare settings, but can also be found in healthy individuals. Staphylococcus aureus is commonly spread from contaminated hands, and the risk of infection is even higher in healthcare facilities. Escherichia coli is typically found in the environment and foods. These strains are among the most common microbial flora found on the skin of humans and cause life-threatening infections. Because of the serious risk associated with these strains and their common occurrence, the practice of good hygiene and use of efficient methods of disinfection is imperative. The methods of disinfection were similar across many studies with varying differences. The most commonly agreed upon method of disinfection is surrounded the use of alcohol, whether it's ethanol or isopropyl alcohol. Concentration levels between 60 and 95% are most effective, as this range has higher disinfection properties. Alcohol is generally used for disinfection in many areas of life, mainly because it is bactericidal, virucidal, and fungicidal, as well as safe for common use. Monterv et al. Discussed sodium bicarbonate as a chemical used for cleansing. However, compared to other chemicals, it is lower in toxicity, which makes it safer for frequent or even everyday use. The researchers highlighted its ability to loosen debris at an efficient rate without the harmful risks other chemicals pose. One review discussed that soap-based cleansers were more harsh on the skin and cause more irritation than the synthetic ones. Cleansers that are safer but still have effective antimicrobial properties are preferred and could allow for prolonged usage of cosmetics. Many studies highlighted that disinfection alone will not efficiently stop the spread of bacteria, thus highlighting the importance of good hygiene. The importance of proper hygiene was a commonly discussed factor in preventing the spread of bacteria and general illness. One review stated that hands transmit more than 80% of human illnesses. Since our hands are used for the application of many products, hand hygiene is necessary in preventing and controlling the spread of bacteria and those illnesses. It is mutually agreed upon across multiple studies that consumers' hygiene habits greatly impact the risk of bacterial contamination. Bacteria can also be spread to and from the face. When the bacteria from our face come in contact with the products we use, a buildup of bacteria can occur. Since the use of harsh cleansers pose a risk to skin health, methods of maintaining clean products are useful to know. Hygiene can also impact the longevity of the makeup products, which means the better consumers' hygiene is, the longer the products are expected to last and function at the expected standard. This led me to question ways bacteria could be eliminated in cosmetic applicators. Specifically, I aim to answer the following. What is the most effective method at reducing the transmission of bacteria in disposable makeup sponges? This led me to developing my hypotheses. My research hypothesis was, if 70% isopropyl alcohol is used to clean disposable makeup sponges, then bacteria growth will be significantly reduced compared to sodium bicarbonate and hydrogen peroxide. Therefore, my null hypothesis was, if 70% isopropyl alcohol is used to clean disposable makeup sponges, then there will be no difference in bacteria growth among the different treatments. My independent variables of my experiment were 70% isopropyl alcohol, hydrogen peroxide, and sodium bicarbonate, with bacteria growth being the dependent variable. As a control, I used distilled water. The lab environment, incubation temperature, and incubation time period were held constant. The participants were all administered the same type of sponge for the experiment, and the amount of time the sponges were soaked in each treatment was also held constant. I began my experiment by obtaining informed consent from administration, the participants, and their guardians. After I had enough participants, I used an online random generator to choose 10 people. The sample size of my experiment was 40, and the sample population included the disposable sponges. I then gathered all necessary materials for my experiment and collected bacteria from my participants via the makeup sponges. I distributed 4 storage baggies with 1 makeup sponge in each to all of the participants. The bags were labeled with the number 1 through 10 for organizational purposes. The participants were sent a detailed outline on how to use the sponges and for how long. They used the sponges for exactly 7 nights, and then I collected the materials to begin my data collection. Each participant set of 4 sponges inoculated 1 petri dish. I labeled the petri dishes in correspondence with sponge number and treatment. I did this by labeling them with the number 1 through 10 and the letter lowercase a for before treatment or the letter lowercase b for after treatment. I then inoculated the dishes with the makeup sponges before and after receiving the treatments. Each group of sponges were soaked in their respective treatment for 3 minutes, and then the excess liquid was squeezed from them and laid to dry on the paper towels while the next set of sponges were soaking in the treatment. The petri dishes were then placed in the incubator at 37 degrees Celsius for 24 hours. I then compiled my raw data, which is what the picture above depicts. I was tasked with making an original chart to assign the petri dishes a percentage of growth, since there was no definitive way to measure the bacteria growth. I then conducted a one-way ANOVA statistical analysis. I ran a one-way ANOVA test to determine any significance between the groups. At the 0.05 significance level, the null hypothesis was rejected with a p-value of less than 0.001. This is a bar graph that best represents the data from the one-way ANOVA test. It's a cluster bar graph that shows the sum in blue and the average in orange of bacteria growth for each treatment group. The different sums among the treatment groups show which groups have the highest percentage of growth. The averages show the average percentage of growth in each group. In this chart, it can be clearly seen that the group that received the hydrogen peroxide had the lowest overall percentage of growth compared to the rest of the treatments. The chart also displays the effect isopropyl alcohol had on the bacteria in the sponges. The bacteria on the sponges was reduced after being soaked in 70% isopropyl alcohol, and the chart suggests that there is enough evidence to conclude that the reduction was significant. After the one-way ANOVA test determined any significance between the groups, I then wanted to determine which group was the most effective at reducing bacteria growth. Since the results showed that the hydrogen peroxide was the most effective, also significantly reduced the bacteria growth, I used a t-test to determine the most effective group, either 70% isopropyl alcohol or 3% hydrogen peroxide. This information in the above chart conveys that the hydrogen peroxide was the best treatment at reducing bacteria growth. It can be seen that the isopropyl alcohol treatment significantly reduced bacteria growth, just not as well as the hydrogen peroxide. So, while I was able to reject the null hypothesis due to the significance of the results, isopropyl alcohol was not the best method at reducing bacteria growth. I was successfully able to reject the null hypothesis that if applied to the sponges, 70% isopropyl alcohol wouldn't make a difference compared to the other treatments. In conducting this study, I was also able to find the best method at reducing bacteria growth in makeup sponges, thus answering my research question. The results of this study imply the importance of good hygiene in terms of makeup products. This study specifically surrounds the importance of keeping makeup applicators bacteria-free, or at least reduced as much as possible. Applying the results to everyday life, most of us know that makeup applicators need to be cleaned at some point. However, the cleansers available on the market aren't great at reducing bacteria growth. The cleanser might be removing a buildup of makeup, but bacteria will still be present if not treated with an antibacterial cleanser. Therefore, in addition to a normal applicator cleanse, adding a hydrogen peroxide soak could significantly reduce bacteria growth. This will limit chances of infection from the bacteria being transferred to your face. Some limitations that impacted my study included my inexperience with cultured mediums. When I was inoculating the petri dishes, some of the quadrants were not covered in their entirety, and this led to the reason why so many of the samples had only 90% growth. It can be seen in the picture that the growth does not reach the corners of the quadrant. However, where it was inoculated, it does have 100% growth. What also could have limited my results was the reuse of treatments. The treatments were not emptied and refilled after each participant's sponges had soaked in them. This could have led to cross-contamination and swayed the amount of bacteria growth. However, it is important to note here that the sponges that were soaked in hydrogen peroxide had consistent results in all except one sample. Another limitation of this study is the limited time available to complete multiple trials. If there was more time to complete multiple trials in this study, the reliability and validity of the results could have been stronger. During my preliminary research, I found that isopropyl alcohol was the main solution or treatment being tested and applied in research. Other researchers did have success in those studies. However, it can now be taken into consideration that a better method is out there. When considering the effectiveness of isopropyl alcohol at killing bacteria, researchers should also consider that hydrogen peroxide may work better. Future studies could perform research on the effect hydrogen peroxide in other fields when killing bacteria is a focal point. Different concentrations could also be tested against bacteria growth in other areas of study. Because of these findings, I'll be implementing the results into my own life. I suspected that just because there was no makeup on my applicators, that there was probably still a buildup of bacteria somewhere. In addition to using an applicator cleanser to remove makeup, I will be soaking my brushes and sponges in hydrogen peroxide for at least 3 minutes, such as I did in my own experiment. Through this study, I have developed a better understanding and appreciation for the research process. My time management skills have also improved from this process because procrastination was not my friend, and I've learned a lot from this experience. I would like to thank everyone here who helped me with this research project. My teachers here at PGS, specifically Ms. Long, for helping me through the entire research process, from developing my research question to helping me in the lab, and Mrs. Dillon, who helped me develop my methodology and helped me with my raw data. I would also like to thank Kim Magner and Dr. Bocock for allowing this research to take place. A big thank you to the participants in my study. Their participation allowed me to work with human bacteria rather than having to purchase it. They took the time to get me what I needed for my research and got it back to me in a timely manner. Again, I just want to say thank you to everyone who contributed to this process. Thank you, and at this time, I'll take any questions. Yes, Ms. Probst.
For your control, if you can clarify for me, was the control sponge actually used for makeup and then cleaned with distilled water, or was it just a plain sponge that had not been used for makeup?
Ms. Probst is asking if the sponges already had makeup on them before using the control for the treatment, or if they were just plain sponges. To answer your question, all the sponges were just plain sponges. Some of the participants did have makeup on, so there was still residual on there. However, most of the participants did not have makeup on, so it just kind of varied, and that could just be another limitation of my study.
Okay, I was just wondering how you account for, the sponges are not sterile from the manufacturer, so the sponge itself could have bacteria on it and not just the person.
Right.
So that could be for future studies.
Okay.
So when you did your literature review and you looked at alcohol, you said that most of the studies focused on the effects of alcohol. So why do you think the focus is on alcohol versus hydrogen peroxide?
Dr. Jones is asking why the, I'm sorry, could you repeat that?
Your literature review, I think you said that most of the studies focused on alcohol.
Yes.
To determine the effectiveness of that, but not hydrogen peroxide. Do you have a thought as to why that may be the case?
Okay, so Dr. Jones is asking why the focus was on alcohol rather than hydrogen peroxide. So I think the reason why there was so much focus on alcohol is because I was specifically searching for the effects of alcohol rather than hydrogen peroxide. So those were the main studies that were taking place in my literature, and I think there's just limited research on hydrogen peroxide on the effect of makeup applicators in general. Yes, Mr. Hare.
So this is not a space that I know well in makeup, but I'm gonna try, so if I ask a crazy question, then you don't have to answer it. So your goal was to reduce the bacterial contamination in these applicators, correct?
Yes, sir.
So to Mrs. Prost's point, the sponge is sterile when it comes out of the package. So why do you not use a new sponge each time that you put makeup on?
So Mr. Hare is asking why, since the makeup sponges come out of the bag sterile, why don't people clean their sponges after each time they wear makeup? So I think it's just a matter of convenience. Not everyone has the time to wash their applicators each time they wear makeup. So I think there's also just a lack of knowledge surrounding like what could be on their makeup applicators. So yeah.
Thank you.
Yes, Mr. Shaw.
Do the manufacturers of the sponges typically say use their sponges only once? And you're saying that typically they're used more than once?
Mr. Shaw is asking if the manufacturers suggest using it only once or if it's repeated use. So it does not specifically say, so I guess people just assume they can use it multiple times, and I think it's just preference, personal preference, as to when they choose to use a new sponge or a new brush or whatever.